Okay, so we are currently in the process of discussing the development of cancer. Okay, so we were looking at a few bits of nomenclature at the moment. So we're looking at the classification of different types of cancer into the main two classifications of carcinoma and sarcoma, and now we're looking at types of cancer which don't fit into either of those. So we've talked about leukemias and lymphomas, which are both cancers of the white blood cells. Okay, another example of a cancer which doesn't conform to either carcinoma or sarcoma is cancers arising from nervous system tissue. So cancers arising from tissues of the brain, for instance. So brain tumours are one of the most dreaded forms of cancer of them all. Okay, they're actually extremely rare to actually get a cancer formed from brain cells, but they're not unheard of. So cancers arising from nervous tissue. Okay, right. So that doesn't conform to either of those classifications either. Okay, so a few more pieces of terminology. These are all words: carcinoma, sarcoma, leukemia, lymphoma. These are all words for cancer. Okay, there are a few other pieces of terminology I'd like to mention that refer to benign tumours rather than actually malignant tumours. So they're not actually nomenclature referring to cancer, but they're referring to the forerunners to cancer, and then they have corresponding words which then refer to the malignant tumour. Okay, so there's another piece of terminology which is you'll often hear in the cancer literature, which is adenoma. Okay, so what is an adenoma? An adenoma is a benign tumour. Okay, so these pieces of terminology I'm about to mention, these are all benign tumours. So an adenoma is not cancer yet, it's the forerunner to cancer. Okay, it's that point where you've got a large new mass of cells, but at this stage they have not acquired the necessary mutations. None of the cells in that mass have acquired the necessary mutations for them to start moving around, so they haven't lost their positioning yet. Okay, so they're not invasive. Okay, so an adenoma is the name for a benign tumour in a glandular, in a glandular epithelial tissue. Okay, so glandular epithelial tissue, benign tumour of a glandular epithelial tissue. So what examples do we have of glandular epithelial tissues? Well, we've seen loads of these now. So for instance, the gastrointestinal tract, the lining of the gastrointestinal tract, all of that is pretty much a glandular epithelium. Okay, you've always got little secretory glands all the way along the gastrointestinal tract which are secreting lubricants that are helping to move the food boluses along. Okay, so the gastrointestinal tract is one huge great gland that's secreting stuff. It's an exocrine gland, it's secreting stuff onto the outside. Okay, then we've also seen the pancreas, that's an exocrine gland, it's secreting stuff into the intestine again. The liver is a huge great exocrine gland, that's again an epithelial tissue. Okay, the lungs, the lining of the lungs, they're exocrine glands as well. So all of these sorts of epithelia, these can form adenomas. Okay, now there is a corresponding word to describe a malignant tumour of a glandular epithelial tissue. Now you could describe it as a carcinoma, but if you wanted to be even more specific, the malignant form is an adenocarcinoma. Okay, so if you want to tell the person that you're not just talking about any old epithelium here, you're talking about a glandular epithelium, then you'd call your carcinoma an adenocarcinoma. Okay, so this is the malignant tumour. So this is the forerunner, the benign tumour, underlined in green here, and this is the malignant tumour. Okay, so adenoma and adenocarcinoma, those are two pieces of terminology that you can't really avoid in the cancer literature. Okay, just two more now, which are chondroma and chondrosarcoma. So chondroma is the benign tumour, and then chondrosarcoma is the malignant tumour, the cancer. Okay, so what is this? Well, sarcoma tells us that this is a connective tissue cancer, a cancer arising in connective tissue. The chondro tells us that it's a specific type of connective tissue. Now, chondro means pertaining to cartilage. Whenever you see the prefix chondro, it means that whatever follows that, it's something to do with cartilage, basically. Okay, so this is a sarcoma in cartilage, and cartilage is a form of connective tissue. Okay, so the initial benign tumour is called a chondroma. The malignant tumour that you then get from the benign tumour, if you're unlucky enough, and I do want to stress that, you know, it takes a long time to go from having a benign tumour to having a malignant tumour. It's not something that occurs overnight. Okay, it's a long process waiting from developing a benign tumour for it to become malignant. Okay, but when it does become malignant, it's then called a chondrosarcoma. Okay, so those are just a few pieces of terminology. Now, before we go on to a more in-depth model for what cancer is and for how it develops, what I want to talk about is a really important point, which I'm sure you're probably already familiar with, but I just want to make sure that everyone understands this, which is that a cancer cell is not just a single type of cell. Okay, so if you have a liver cancer cell and a skin cancer cell, those are not the same type of cell. Okay, so cancer cells are different from different types of cancer. So if you've got a skin cancer, those cancer cells are very different from liver cancer cells. Cancer is not one disease, is what I'm trying to say. And even if you've got this, you know, if you've got two people with the same type of cancer, so they've both got, let's say, a form of skin cancer, those two cancers can be very different. Okay, the exact mutations that you have to get in order to make the cell over-divide and also to make the cell mobile, these can vary between different types of cancer and also between different cases of the same type of cancer. Okay, so it's a very complex disease, cancer. It's not one disease that occurs in every single person, okay, in every single type of cancer and in every single patient. It's a disease that varies very much so depending on the location that it's happening, and also even if you're having it occurring in the same location in two different patients, it can be very different, basically. Okay, so it's a complicated disease. There is not just one mutation which makes you over-divide, which is shown in all cancers. There's not just one mutation that then makes you metastatic, which is shown in all tumours. Okay, and to demonstrate this point, I'm going to tell you about two types of skin cancer, okay, to demonstrate how different they are. So, we'll start with basal cell carcinoma. Okay, now it's a type of skin cancer, so carcinoma tells us that we're dealing with an epithelium here, which indeed the epidermis is. Okay, and basal cell, what does that suggest? Well, we know something about the skin. We know something about the epidermis. There is a basal layer of the epidermis, and this is exactly why this is called basal cell carcinoma, because it actually occurs in the basal cells. In fact, basal cell carcinomas are suspected to arise from epidermal stem cells. So let me just redraw out that picture of the skin that we drew. Okay, so here are the three layers of the skin. So remember, the basal layer of cells of the epidermis here contains epidermal stem cells. Okay, now in basal cell carcinoma, the cell that originally goes wrong, the cell that originally gets that change that then allows it to over-divide, producing a population of cells which all over-divide. And by the way, a population of cells all over-dividing is then called the clone of that cell. Okay, and then within that clone, within that population of these cells that are all over-dividing, you then get one of them. You just need one of them at some point to get another mutation. Well, it's more complicated. We're about to come on to a more complicated model, but in my initial very simple model, you then just need another one to get another mutation, which then allows it to move around freely, and then you've got an invasive malignant tumour and you've got cancer. Okay, so in basal cell carcinoma, the cell that originally goes wrong is believed to be the an epidermal stem cell, basically. Okay, and I say it's believed to be an epidermal stem cell. We don't know for sure what the origin of basal cell carcinoma is. Okay, there's a huge number of things we don't know about cancer. Okay, and this is one of them. In basal cell carcinoma, we suspect that the starting cell, the cell that originally goes wrong, is some epidermal stem cell, but we don't know, basically. Okay, right. Now, what was my point to this? Oh, my point was that different types of skin cancer can be very, very different. So, let me contrast this to another type of cancer, which is a melanoma. Okay, so also in the epidermis, you have cells known as melanocytes. Okay, and these are completely different from the cells which originate from the epidermal stem cells. Okay, so also in the epidermis you will have melanocytes. Okay, and these cells can produce melanomas. So let me draw a melanocyte. So these cells produce the pigment, okay, melanin, and they are responsible for you getting a tan, basically. When you go out in the sun, these cells will produce more of the pigment melanin, okay, and that will protect you from ultraviolet radiation. Okay, now in melanoma, the cell that originally goes wrong is a melanocyte, and it starts over-dividing, producing a huge, great population of melanocytes here that are all over-dividing, and then one of these gets a further mutation and then becomes invasive and metastatic. Okay, now, my point to this was that in basal cell carcinoma, the cancerous cells all have properties similar to the keratinocytes, the main cells of the epidermis. Okay, and the key property of keratinocytes is that they have a huge amount of keratin. So the cancer cells of a basal cell carcinoma contain a huge amount of keratin. Now let me just give you some idea of what keratin actually is. Keratin is a protein filament that is a very important component of the cytoskeleton. Okay, so basically, although we draw pictures of cells that look like this, okay, this is kind of our nice simple picture of a cell, it's inaccurate. Okay, we draw the cytoplasm as this sort of nice empty gloop, basically, but in reality, the cytoplasm is not just some gloopy mush. Okay, instead, the cytoskeleton is full, absolutely full of a meshwork, a very rigid protein meshwork, loads of little protein filaments going everywhere, basically. Okay, you've got this meshwork, it's like a very, very dense spider's web, and this is the reason cells have a rigid structure. Okay, you might wonder, why do cells stay in the same shape? Well, it's because they have this rigid structure that is holding them in the same shape. The cytoplasm is gloop. Okay, the cell membrane, that's gloop as well, but what's holding the cell in the same structure is that you have this rigid frame that is holding the cell membrane up, and that's why cells stay in the same shape. Now, this rigid frame is known as the cytoskeleton, which is quite appropriate because skeletons hold things up. Okay, cyto means pertaining to cells, skeleton means a rigid structure holding something up. So this is the cytoskeleton, okay, and I'll colour a bit of it in here. So this is filaments all over the place. Now, one of the components of the cytoskeleton that is overrepresented in these keratinocytes is keratin. Okay, so most cells have some keratin, but it's not a major component of the cytoskeleton, not compared to actin. Okay, but in these keratinocytes, in the cells of your skin, you have a huge amount of keratin. Now, keratin is a protein filament. Now, what does that mean? It means that to build a protein filament, to build a keratin filament, you have to take loads of individual keratin proteins and stick them together. Okay, so it's a huge, great fiber, basically, and it's not a single protein, it's loads of these little proteins all stuck together. So these are the individual keratin proteins that I'm trying to represent here in orange. Okay, I'm not going to go into detail of how exactly they aggregate together to form this filament, but the basic message is that to build a keratin filament, you join together many keratin proteins to make a huge aggregation which forms like a fiber. Okay, and these are the fibers that are contributing to this cytoskeletal meshwork that holds the cell in a rigid structure. Okay, right. So in these basal cell carcinomas, these cells, the cancer cells, still have a huge amount of keratin. Okay, the melanocytes are very different from keratinocytes. What are they full of? They're full of this melanin. So the melanoma cancer cells, these end up full of melanin, not always, but usually. So usually melanomas appear very, very dark because of all the cells are all full of melanin. Okay, so that example was meant to illustrate the fact that the cancer cells of the melanoma are very different from the cancer cells of the basal cell carcinoma. These ones are full of keratin, these ones are full of melanin. Okay, so there is not just one type of cancer cell. Do not think that you start off with a cell of a tissue and then it has to go through some stages to become this type of cell known as a cancer cell, and that no matter what the starting tissue is, if you go through these changes you arrive at the same type of cell. It's not like that. Cancerous is a very broad property that many, many very different cells can have, basically. It's this property that you are being incredibly selfish, you've got this antisocial behaviour, you just want to propagate your own progeny, and you don't care about the multicellular organism. That's what cancer is, and many different cells can have that property. Okay, right, so now what we're going to turn our attention to is the more in-detailed model of how cancer forms, okay, and some of the other mutations that you need to acquire in order to actually become cancerous, okay, because my initial model where you just needed two, that is completely oversimplified. Okay, so we'll go for this again then. I think we'll do it in a duct again. The duct is a nice example. Right, so here is our duct once again. Here are our healthy duct cells at the moment. In fact, the whole duct is healthy. We'll start from the healthy duct. So here are these epithelial cells lining this duct, and then I'll put in the basal lamina on which they all sit here in turquoise. Right, so we start off exactly the same as before with a single cell that is going to go wrong. Okay, so a cell that's going to go wrong. We'll say it's this orange one up here this time. So this is the cell that's going to go wrong this time. Okay, and it starts off by acquiring some heritable change in usually some in some protein that's responsible either for stopping the growth pathways or for activating growth pathways. Okay, so remember, you need some change that's going to cause this cell to overgrow. So the first initial step is the same as before. We're just going to add in more steps. So this cell is going to get some heritable change that means that it can overgrow. And that's either a gain of function, GOF, in the growth inducing pathways, so in the pro-growth and division pathways. Okay, pro-growth and division pathways, or it's a loss of function in the pathways which inhibit growth and division. Loss of function in anti-growth and division pathways. So something that tips the balance and allows this cell to divide faster than its neighbours, basically. Okay, and this gives it a selective advantage if we're talking about this Darwinian-like microevolutionary process. Okay, so now what's going to happen then is that you're now going to get this cell proliferating. So let's draw this happening. So we've got some new orange cells here, and these new progeny of this original cell, these are all dividing too quickly as well. So what you'll start to get is our little lump. Okay, so you're already starting to get a little benign tumour there. Okay, now it might not be visible initially, in which case it wouldn't be diagnosed and it wouldn't be called a benign tumour. If you get a very, very powerful mutation, okay, so for instance, potentially an activation of a protein known as MYC. There is a very powerful protein within cells called MYC. I'll just write this down. MYC. okay, which is an incredibly powerful protein at making cells divide. It's often called the cell's most powerful mitogen. Cell's most powerful mitogen. Okay, so if you get some sort of gain of function in MYC, maybe you produce far too much MYC, okay, maybe that was caused by an epigenetic change, or maybe a mutation in the promoter region of MYC. Many things could cause a rise in the level of MYC, or maybe you get a mutation in the MYC that makes it even more active, or maybe some problems with the destruction pathways for MYC. So there are many things that could cause this. Cell's most powerful mitogen is what MYC is often called. It's given that grand title. Okay, so if you get a very, very powerful initial mutation, then this might be capable of giving you a visible new growth, a visible neoplasm basically, and in that case it would be called a benign tumour. So this initial stage is only going to be called a benign tumour if it's actually seeable. If it's so small that you can't see it, then it's not going to be labelled a benign tumour. Okay, right. Now, I then said that that was the first stage, and then the second stage was now you just need one of the cells in this benign tumour to then get a mutation that means that it's invasive. Okay, in reality there are more steps that I now want to put in. There are many other properties that need to come first. Okay, but the basic principle remains the same, that you initially get this first mutation, and we do believe that the first mutation is generally a mutation that causes overdivision, okay, to give this population of cells that are all over-dividing. Then, what's going to happen is in one of these cells in this clone, okay, of cells which are all capable of over-dividing, another mutation is going to occur. So that's just pick one at random, because it will be random. Okay, so you now need another mutation, and this is the basic principle of cancer, that it's a very gradual process that occurs over a human lifetime. Okay, you are gradually producing this clone of cells that is gradually evolving. Okay, they're gradually going to get more and more mutations, and they're going to get closer and closer to becoming cancer. Okay, so let's see this. So this cell here that I've outlined, it's now going to get another mutation. Okay, that's going to give it another property that is necessary for cancer. Now, this one is not yet going to get that invasive property. Okay, there are other properties that you need first. So generally, a very early property that you acquire is something known as genetic instability. So we can imagine that this is the second mutation that occurs. Okay, it would certainly be a good model to imagine that it's the second mutation that occurs. Whether it actually always is another matter. Okay, so genetic instability. It's certainly true that most cancers at some point gain genetic instability. Whether it is the second mutation or maybe the third or the fourth, that's obviously a different question, or maybe the first even. Okay, so what do I mean by the property of genetic instability? Okay, so genetic instability means that this cell is going to be much worse off as far as gaining mutations is concerned. Okay, so the cell's ability to get mutations is going to hugely increase. Okay, that's the property of genetic instability. Now, how can this occur? Well, if, for instance, you got mutations in the mechanisms underlying DNA repair, okay, so cells actually get mutations pretty regularly, far more regularly than you would think, okay, and the usual thing that saves the day is that you have a huge number of repair mechanisms that repair the mutation, and there's very clever mechanisms for doing it, okay. If you got mutations that meant that that the proteins involved in DNA repair could no longer function, then you would stop repairing those mutations, and they would last, basically. They would truly become mutations, and then that would mean that this cell's ability to gain mutations would be much, much increased. Okay, that is what is meant by genetic instability. Okay, so potentially it's due to problems with DNA repair mechanisms. Another thing that can often happen is that they can get problems with segregation in mitosis. So let me just describe to you the process of mitosis. So segregation in mitosis, which is particularly severe because these cells are dividing so fast. Okay, so let me just remind you of the cell cycle. Okay, so in the cell cycle, you start off, the first phase of the cell cycle is a phase known as G1 phase. Okay, so we'll have this here. So this is called G1 phase, and this stands for the first growth phase, or the first gap phase as well. Okay, you can either view the G as standing for growth, or you can view it as standing for gap. So this is the first growth/gap phase. Okay, now in this phase, what is happening is the cell is growing. So remember, long ago now, I defined for you what the difference between cell growth and cell division was. Cell growth was the actual individual cell getting bigger. Okay, so in this first growth phase, what happens is the cell goes from, let's say, being this size, okay, here, to being maybe this size. Okay, right, so it has grown basically. Now, in addition, in the first growth phase, what it's going to get ready to do is copy its DNA. So this cell is going to divide. It's going through the cell cycle. Okay, so it's going to divide. So in order to divide, it needs to copy all of its DNA, because both of the daughter cells need to have a copy of every single one of the 46 chromosomes. Okay, now cells do not usually just have the enzymes and all the machinery needed to copy DNA just sitting around. Instead, they synthesize them when they are needed. So in this first growth phase, you're going to be making all of the necessary components for replication of the DNA. Okay, then you're going to go into the phase where you actually do replicate the DNA. So you're now going to go into the phase known as S phase. Okay, and in S phase, what's going to happen is you're going to copy every single one of the 46 chromosomes. So I'm going to draw the cell much bigger now because I want to actually show the chromosomes. Okay, so remember, in the nucleus you have 46 chromosomes, 23 pairs, and now what you're going to do is copy every single one of them. So let's just show one of them here. Okay, so here's one chromosome. Okay, now it's going to get copied, so you're going to make an identical copy of it, and this will be happening for all 46 chromosomes. Okay, and these two, which are truly identical, these are now known as the sister chromatids. Okay, here. So in S phase you copy the DNA, okay, but you have not yet split the nucleus. Now what you have is a massive great cell with two copies of every single one of the chromosomes in its nucleus. Okay, so that's the S phase, which stands for the synthesis phase of the cell cycle. Okay, then the next phase, the third phase of the cell cycle, which we'll have here, is the G2 phase. Okay, and this is the second growth phase. Okay, now in this phase what's going to happen is the cell is going to get even bigger, so it's going to grow even more. Now why is it growing? Well, I explained this earlier, that the cell needs to have enough proteins, have enough intracellular organelles now for two cells. It needs to duplicate all of the essential enzymes in the respiratory pathways. It needs to duplicate all of the receptors on the cell membrane. It needs to make the cell membranes bigger. It needs to make all of the signaling proteins involved in the many intracellular signaling cascades. It needs to make more cytoskeletal components. You know, this is what it's doing in the G2 phase. It's growing so that it's got enough of these things for two cells rather than one cell. In addition, it's also getting ready for the M phase of the cell cycle, which is the final phase of the cell cycle. Okay, so this is M phase, which stands for mitotic phase. But in fact, M phase consists of two separate portions. It consists of mitosis itself, and then a separate process known as cytokinesis. Okay, now, mitosis is not division of the cell. It is instead division of the nucleus in a certain way. Okay, it refers to this nucleus which has two identical copies, importantly, of all 46 chromosomes splitting into two separate nuclei which each have one copy of all 46 chromosomes. It does not refer to the splitting of the cell. Cytokinesis refers to the splitting of the cell. Mitosis, strictly speaking, means nuclear division to produce two identical nuclei. Okay, so in mitosis, what's going to end up happening is by the end of mitosis, you will have two nuclei which are both identical, which each have one copy of all 46 chromosomes. By the end of cytokinesis, you will then have two cells which each have one nucleus, which each have the nucleus which contains one copy of all 46 chromosomes. Okay, right, but cytokinesis and mitosis are both part of M phase. Okay, so even though it's called the mitotic phase, it does contain cytokinesis as well. Okay, so there are only 4 phases of the cell cycle. G0, some people occasionally put G0 as part of the cell cycle. It's not an active component of the cell cycle, so it shouldn't really be viewed as part of the cell cycle. Okay, so segregation in mitosis. So mitosis, this process by which you go from having one cell here with one nucleus which contains 2 copies of every single one of the 46 chromosomes to having 2 nuclei, each with one copy of all 46 chromosomes. How does that occur? Well, basically what happens is the nuclear envelope breaks down, okay? And then what happens is that the sister chromatids, all 46 pairs of sister chromatids, are pulled apart. Okay, so let me get some more paper and show this. So, what's going to happen is we'll start off with our cell here, with its nucleus here, and inside this nucleus we have all 46 pairs of sister chromatids. So here's, let's say, one sister chromatid pair, here's another sister chromatid pair. They could be for both chromosome 1s, for instance. Then you'd have the 2 sister chromatid pairs for the 2 chromosome 2s, and you'd go on. Okay, what will happen is the nuclear membrane will break down. Okay, so here is the cell membrane, and now the nuclear envelope's gone. Okay, then the sister chromatids all line up down the middle, and then what's going to happen? We've only got 4 of them, but in reality there would be 46 of these things here. Okay, so it would go on and on all the way down here to 46. Okay, and then what's going to happen is basically you're going to use protein filaments known as microtubules to pull the sister chromatids apart. Okay, and one copy of every single one of the 46 chromosomes will go to one side of the cell, one pole of the cell, okay, and the other identical copy of all 46 chromosomes will go to the other side of the cell, okay, and then nuclear envelopes will reform around these 2 separate groups of 46 chromosomes to create 2 identical nuclei. This is what is meant by mitotic segregation, okay, of the chromosomes, because we are segregating the chromosomes into 2 groups, into 2 identical groups. Okay, if things go wrong with this, what can happen is that you can end up with nuclei which do not contain one copy of all 46 chromosomes. Let's imagine that something went wrong with this microtubule, okay, and it didn't attach to this one. Then what would happen is when this microtubule contracted, it would pull both of these sister chromatids here to this side, and this nucleus would end up with 2 copies of that chromosome. That is going to cause a humongous great genetic change in that daughter cell that ends up with 2 copies of that chromosome. Okay, it's going to effectively cause an aneuploidy. Okay, most aneuploidies that occur at a germ cell level are embryonic fatal. Very few of them are survivable. Down syndrome is very, very rare in that it's one of the few aneuploidies that is actually survivable. It's trisomy 21. It results because of you having 3 copies of chromosome 21. Most aneuploidies are embryonic lethal. This is happening in a single cell of a single somatic cell, so it's not as bad, okay, as having it at the embryonic level. But this is going to cause a major change to that cell's behavior, and it could lead to cancerous mutations. Okay, right. Well, cancerous behavior in the daughter cell that ends up with more chromosomes, basically. Okay, so this is another way that you can get major changes to DNA, and we're not now talking about just one nucleotide base pair changing, we're talking about a whole new chromosome, okay? So, such extreme types of changes are known as changes to the karyotype, okay? So, karyotype changes. So, karyotype means which chromosomes you have, basically, in your nucleus. So, usually we have 46 chromosomes, and that's our karyotype. Well, 22 pairs of autosomes and then 2 sex chromosomes, that's our karyotype. If you get changes to that, then you've got karyotype changes, and these are very, very severe genetic changes. Okay, in addition, I want to stress that the problems with DNA repair, these can result in major mutations as well. I'm not now just talking about a single base pair being changed. Okay, we're talking about much more severe mutations. Okay, so often when I say mutations, people will assume that I mean things like, if we've got a piece of DNA here, okay, things like a single one of these base pairs being switched. So potentially if I took this CG base pair here and replaced it with a TA base pair, they think that that was what I meant by a mutation. That is a mutation. Okay, it's not a particularly severe mutation. There are scarier mutations that can result from DNA repair mechanisms going down. Okay, so let me describe some of these more severe mutations now. So, when DNA repair mechanisms go down, you can end up with huge pieces of chromosomes being deleted. Okay, so if I draw a chromosome here, so remember, a chromosome is fundamentally just a huge, great piece of double-stranded DNA. Okay, so here's my double-stranded DNA duplex. Okay, so you can end up with huge portions. Let's say this huge, great fragment is just going to get deleted. Okay, that's one of the things that can happen if certain DNA repair mechanisms are down. In addition, you can get duplications. You can start potentially getting another one of these added in earlier on. So instead of deleting it, what you could end up doing is ending up with a new chromosome. Let's say split this chromosome here and stick in an exact replica of this in here. Okay, so you can start getting huge changes to DNA. Okay, so here is this chromosome now. Here is a new one of these orange sections here. And now we've got the original orange section down here. Okay, and we've now got a much bigger chromosome. So basically, when you actually look at the chromosomes of cancer cells down the microscope, you see huge changes. The chromosomes look a complete mess often, okay? And this is originally how people described genetic instability. It was something that you could see down a microscope. The chromosomes were a mess, basically, in these cancer cells, okay? Now what we understand it as is that there are problems with DNA repair mechanisms, potentially problems with mitotic segregation, which are leading to these big scale changes in the chromosomes. Okay, so this is duplication I've described here. Another big change that you can get is what's known as translocation. So let me just now describe translocation, and this is going to be important when we look at chronic myeloid leukemia. The first mutation that occurs in chronic myeloid leukemia is a translocation, basically, a vast translocation. Okay, so we'll see that later. Okay, so translocation, what this means is that you take one chromosome here. Okay, I might as well build this up for chronic myeloid leukemia. So let's say this is a chromosome 9, okay, and then we take another chromosome here, which is chromosome 22, and we'll come back to these specific chromosomes here, okay, and let's say we take a large portion of chromosome 9 here in blue, and a portion of chromosome 22 here, okay. Well, actually, let me do this more accurately. The portion of chromosome 22 you're going to take is bigger than the portion in chromosome 9 in chronic myeloid leukemia. Okay, and what we're going to basically do is break this portion off, and stick this portion back onto here, and stick the blue portion back onto here. Okay, so this is because the DNA repair mechanisms are broken, so we're doing a very, very crude way of stitching these back together. The better DNA repair mechanisms are broken, so we're using a very crude way of repairing this, and we're just sticking them back together randomly and hoping for the best. And then what you'll end up with is these new chromosomes, which are formed by merging part of chromosome 9 with part of chromosome 22, like so. Okay, and then merging part of chromosome 22 up here with this tail portion of chromosome 9 here. Okay, that's what is meant by translocation, and again, it's a huge genetic change. Okay, so when I say genetic instability, I don't just mean, a few base pairs are being changed. These are major changes to the chromosomes that you can see down the light microscope. Okay, so what's the significance of the cells becoming genetically unstable in this way? Well, what's going to happen if we go back to our build-up of cancer? Okay, here is our duct here. Okay, we've got our healthy cells here. We've then got our benign tumor that has formed here. Okay, so I'll just finish the healthy cells here. Okay, and they're sitting on the basement membrane, so I'll put the basement membrane in. Okay, and then we said that we started off with this mutation in one cell that caused this overdivision, which caused the beginnings of a benign tumor. Okay, and that caused a little lump that might be visible depending on how powerful the initial mutation was, and some initial mutations can be powerful enough to produce visible lumps. Okay, so it's not trivial, the first mutation. But now we're going to develop further, and we've decided that one cell, which let's say it's this one here, is now going to get a mutation in some sort of DNA repair mechanisms, or potentially the mechanisms responsible for mitotic segregation, or something along that lines, that is going to mean that it now acquires mutations at a much faster rate. Because if you're making a mess of your chromosomes in this way, a mess of your DNA, then you're going to accumulate mutations much faster than a normal cell. Okay, now, what is this cell going to do? Well, it's now going to divide very, very fast because it's already got the mutation that means that it overdivides. So now, this cell that is genetically unstable, which we'll colour in vivid purple here, or just a black smudge there, okay, this cell is now going to divide and produce a clone of cells that are all extremely genetically unstable. So let me show this. So, I'll just show the cancer now. Well, sorry, it's not cancer yet. I'll just show the tumour now, rather than having to draw the duct out each time. Okay, so, here is this little clone of these cells that are now further along in this process than the orange cells. So we've still got all of the orange cells, but we've now got a new clone of cells that we're more interested in. Okay, right. So this inner little clone of cells here, these are the purple cells. These are the ones which have two mutations now. One is the one that makes them overdivide, okay, and then the other is the one that now makes them genetically unstable. So you now have a population of cells that are going to be accumulating mutations extremely rapidly. Now, some of them will get mutations in very important proteins, okay, and those mutations will be lethal to the cell. So let's say one of them gets a mutation in complex 4 of the electron transport chain. That will be lethal, okay, and it will die. Okay, so you'll have a lot of death in here as well. But some of them will just happen now to get mutations that then allow them to go further on in the way to cancer. So this is what you have created. You have effectively created a breeding ground for cancer. Okay, you have got these rapidly dividing cells that are extremely genetically unstable and are just making new pieces of DNA effectively, and seeing what the result is. It's just a breeding ground for disaster, basically. Now, a lot of the cells will be dying because they'll be making mutations that kill them, but some of them will eventually end up getting mutations that allow them to take the next steps towards cancer. Okay, so this is a breeding ground for cancer, basically. Okay, for carcinogenesis, which means the production of cancer. Okay, right. So, let's now talk about some of the other mutations. Okay, so this is one of the mutations that I've now added in my more refined theory. Okay, you might think, so now are we ready? Now are we ready to go to the invasive property? Well, not yet. There are more properties that we need to put in. Okay, so another property, another mutation that is going to occur at some point in the development of cancer is that you're going to get altered metabolism. Okay, and the mutations, by the way, which I'm about to tell you now, I can't really say what the order that they're going to occur in is. So in fact, I should probably now summarize where we're going, and then I'll tell you about these mutations. So basically the idea is now that one of the cells in this purple clone now is going to get a further mutation, and this further mutation will give it another property that is advantageous, okay? That property might allow it to better survive in the conditions of the tumour. And one of the properties that we're about to look at is a mutation that alters the metabolism of the cell, okay, and better enables it to survive in this difficult place, because you've got a lot of cells that are all demanding nutrients. Okay, if you've got a mutation that allows you to better take up nutrients or better process that nutrients in low oxygen conditions, okay, because there are low oxygen conditions in this tumour, okay, then you will be better equipped to survive in this environment, and you will do better, and you'll therefore make a bigger population of cells. Okay, then after that you're going to, within that population of cells, get another mutation in another individual cell which is going to take it on, and this is the microevolutionary process, this idea that this cycle goes on and on. You gradually produce better and better cells which are more and more capable of outcompeting other cells, okay, until finally that cell, one cell in this tumour acquires enough mutations that it has enough properties to be considered cancer, and especially that it starts to become invasive. Okay, it finally acquires that invasive mutation, and then it starts propagating out and setting up new tumours far, far away. Okay, that's the concept of cancer. That's the microevolutionary process, that you're gradually evolving these cells more and more, creating more and more dangerous cells that are better and better able to survive in these difficult conditions. Okay, right, so that's where we're going. And what we now want to discuss is certain mutations that you need to pick up on the way in that process in order to actually be a fantastically good cell at surviving, and therefore to be a cancer cell. Okay, so by the time you actually get to the cancerous process, you will have picked up all of the mutations that we are now going to discuss. Okay, so the two ones that we've discussed so far are that you need to grow too much, okay, and that's generally the first one that you get, okay, and that you need to be genetically unstable. That again is a very early mutation. That's the mutation that allows these cells to then get more mutations, okay, so it too needs to be a very early mutation. So these two are the first ones that you get, and now the subsequent mutations are just going to make you more and more nasty, basically. Okay, and we'll continue this discussion in the next video.